My name is Aristóteles Góes-Neto. I work in the Federal University of Minas Gerais in the microbiology department. And my main research line is in the biology of fungi. In my professional life and research, I have already studied and researched fungi from the middle of the Amazon forest, completely isolated from everything. Now I am currently studying microorganisms from Antarctic and bacteria that was exposed to Mars and space in the International Space Station. And now we are going to an isolated island in the South Atlantic to study the diversity of fungi.
We're interviewing scientists, pioneers, and Bento Lab users that are pushing the boundaries of accessible biotechnology. Today, I'm talking to Aristóteles Góes-Neto, a professor from Brazil who is using metabarcoding to study fungal diversity. This video is brought to you by Bento Lab, a DNA analysis laboratory you can take anywhere.
My last project was in the middle of Amazonia. Imagine 500,000 square kilometers of park. It's one of the biggest in the world. And from the city that we came by air, the airport, we have almost 22 hours of boats inside the Amazon rivers to stay there. So this last project was very fantastic, and it's so good to see that we could travel hours and hours and hours, and the only thing that you can see is forest with any kind or sign of people or any kind of buildings, anything. And we are in the middle of a project. It's an international cooperation project with Germany, Ukraine, and India. And we are now finishing analyzing samples of bacteria that stay in International Space Station. Some of them stay there with in a place simulating the Mars atmosphere and soil exposed there in the International Space Station as some strains of bacteria, and the same strains is staying on Earth in normal conditions, and we sequenced all the genome of this bacteria, and now we are comparing if occurred some kind of mutations in which genes and so on. So for me, I'm completely passionate from what I do, and I love performing research in such very isolated, pristine, distinct or extreme environments. This is fascinating for me, fascinating to understand the whole biology of communities of microorganisms and their interactions.
Can you tell us about where you're going for your next project?
The island of Trindade is in the middle of South Atlantic, between South America and Africa. So it is completely isolated by more than 1,000 kilometers of ocean from one side or for the other side in the direction of the two continents. And we know that from a global biogeographical point of view, studying isolated islands is very important because it's a way that we can understand how the colonization of the island by different organisms occur from the mainland. Our aim is to understand how these spores from the continent, from South America and in Africa, can reach and which species occur in the air of the island and can potentially colonize the soils or the plants or so on and stay there. So it's very important from a global biogeographical point of view.
So what are the scientific methods that you're using?
Well, in the island, there are many different environments there. So we have some parts of the island that we have very interesting kind of forest, relictual forest made of arboreals or tree ferns. That's very, very rare. We have other parts of the island that we have fields with endemic grasses and plants and so on, and some parts that it's the soil the soil coverage is very recent because the island as a whole and from a geographic time scale is very recent, from 3 million years. So we are going to stay completely isolated. We have to carry with us everything, all the device, all the equipment to do the research. And using the Bento Lab is the only way to perform this research, because we have many distinct sites from the distinct types of environment, microenvironment in the island, and we are going to collect soil, samples of soil and samples of the circulating air, and then all these samples we will return to the research station there, and using the Bento Lab we're going to perform the extraction of DNA of everything, and then afterwards, using Bento Lab, we are going to using the thermocycler, we are going to perform the barcoding of the DNA and to the specific regions that with these regions we can, with these amplicons, with the regions, we can sequence using a portable sequencer too from a company.
Is that the MinION?
MinION, yes, from the Oxford, yes, the MinION. And then we can analyze all of these because despite we don't have internet there, we can analyze all of these with a high performance laptop to analyze the billions of sequences that will be generated. And then after the 1 month there, we return to our lab, and with internet we can perform the rest of the analysis.
Thank you so much, Ari, for taking time to talk to me today. It was a real pleasure.
Thank you very much.
You can find out more about Ari's work in the description below. And if you have a question, leave a comment. If you liked the video, give us a thumbs up, and please support us by subscribing. Thanks, and see you next week.